Welcome to today's webinar on nanoscale characterization of lithium metal batteries enabled by cryogenic lift-out. My name is Ben Miller, and I'm the in situ application specialist at Gatan. For the past year or so, I've been working with researchers at UPenn, including Eric Stach and his group, who have been testing our new Metro counting camera. And it was during this collaboration that I met Jun Ko, who will be presenting some of his research today. Jun received his bachelor's and master's degrees in chemical engineering at Korea Advanced Institute of Science and Technology. He's currently a PhD student at UPenn in the Material Science and Engineering Department, working with Professor Eric Stach. His research focuses on characterization of battery materials with cryogenic electron and ion beam microscopy. Jun, if you could go to the slide about the questions now. If you have any questions for Jun during the webinar, you can submit those using the questions pane, as you see here. And after the presentation is finished, I'll try to select a few of these questions to ask Jun live. But if we don't get around to answering your question live, we'll send you an answer via email in the next week or so. So now I'll hand the webinar over to Jun to present some of his recent results.
Okay, can you guys hear me?
Yep.
Hello? Oh,
sounds
okay.
good.
Okay. Hello, everyone. I'm Hyeongjun Ko, a PhD student in Stach group at University of Pennsylvania. Today, I will speak about how cryogenic lift-out enables nanoscale characterization of battery materials, especially for lithium metal batteries. Before we start, I will bring this up first. When I was a first and second year PhD student, it was unfortunately when COVID hit. So I wasn't able to run and train on microscopes. So this was pretty frustrating moment for me. All I could do was watching webinars and reading articles. And when I was asked to give this talk, I felt a little bit bizarre because it brought me back to the moment when I had to stay at my home watching webinars. The webinars I watched were pretty helpful and informative, although I couldn't learn hands-on experience. I hope my webinar is also helpful for your research journey. And it is pretty cool to be able to share my research progress and the pioneering research in this field, and thank everyone else at Gatan for giving me this opportunity. So let's talk about my outline motivation first. I'll speak about why we are interested in lithium metal batteries and how important the cryogenic electron microscopy is. There has been tremendous energy demand, such as electrical vehicles and large energy grid storage systems. In order to achieve such and meet such high energy demands, we need to develop energy storage devices beyond lithium-ion chemistry. Lithium metal batteries are one of the promising ways to increase energy density in batteries by increasing gravimetric capacity 10 times higher than the gravimetric capacity of graphites. And you can also lower the reduction potentials by increasing the full cell voltage. This way, we can improve energy density in the system, as the energy density is the factor of the potential and the gravimetric capacity in the battery system. However, developing lithium metal anodes is still challenging, as the interface in the batteries are not stable. The interface called solid electrolyte interface always form due to electrolyte decomposition. As these interface controls transport of ion and electron, we need to understand what's going on in this interface. As the eye turns out to be as thin as a few tens of nanometers, sometimes it can grow up to 200 nanometer. So as it is nanometer at scale, and we need to use electron microscopy to understand this feature. Okay, here shows why cryogenic electron microscopy plays an important role in characterization of battery materials. When we attempt high-resolution imaging of SEIs, you can easily damage your samples at room temperature, but combined with air-free transfer, now we can attempt high-resolution imaging of battery interface at cryogenic temperatures. And as you can see on the left image, this is a significant research progress because we can see and image SEI at nanometer scale and even at atomic scale while observing the features in the SEI. Okay, let me talk about sample preparation, how we prepare our samples for the cryogenic electron microscopy and their advantage and disadvantage. So most of the lithium metal characterization has been done in the following way. We can deposit lithium on TM grids inside coin cell and specific capacity. And we can open the coin cells to extract lithium deposit copper grids to characterize them. Although this is very facile sample preparation method, there are several limitations. For example, if you target for potassium or calcium batteries, you cannot rely on this method because you cannot get desired electron transparency. The reason why this method works is that lithium is one of the lightest materials, so they can be easily electron transparent. So therefore, we cannot get uniform electron transparency even with lithium metal battery with most cases. So important information adjacent to lithium dendrite can be easily obscured by this method. And finally, deposition and stripping process can be different from that from flat copper foils when you use TM grids as a current collector. Because of the limitation that I mentioned in the previous slide, I believe we need to use cryogenic lift-out process. The cryogenic lift-out is one way to make a cryo-TM sample at cryogenic temperatures. In the focused ion beam microscope, we can trench battery materials and perform lift-out. And finally, we can thin down until we have electron transmission. There are few important advantages in using this technique. First of all, we can preserve as much information as possible when you prepare battery materials. And secondly, you can have desired electron transparency regardless of your atomic numbers of your systems and the original thickness of your samples. However, there are still limitations in this method. First of all, it is pretty time-consuming to make one single sample, and there exist still concerns about ion beam damage. Cryo-TM's preparation at cryogenic temperatures can be done in the following manner. We graphically summarize all this into one slide, but there are a lot of details in each step, but I will briefly go over this. So when you first prepare TM samples in your glow box, we can plunge freeze your sample into liquid nitrogen. During this step, samples are stored inside a plastic bag before it is dumped into liquid nitrogen bath to minimize air contact. And once your sample is on the liquid nitrogen, we cut the plastic bag into half to extract the sample. And you can put your samples in liquid nitrogen working station of the Gatan microsystem, and that the vacuum transfer tool below here is used to transfer the quench sample to the cryo-FIP SEM stage. Once TM sample preparation done, you can take out the sample using the same vacuum transfer tool. Finally, you can store samples on the liquid nitrogen until you perform cryo-TM analysis. The most difficult part in cryo-TM sample preparation is, of course, cryogenic lift-out, because you cannot use selected area deposition like you do at room temperature to weld your sample to the micromanipulator. However, there are several ways to make the approach possible. One way is redeposition welding. In this process, you can mill the resin near where you make a contact between the cold manipulator and your lamella, and redeposition particle will be splashed into the gap, and your sample can be welded within 30 seconds. And this way, you can extract your samples, and you can place your sample to a half grid in the same manner, and you can thin down until you have electron transmission. There are other commercially available ways to do lift-off. First is you can use water vapor to deposit ice everywhere to weld your sample to the cold manipulator or half grid, or you can use cryogenic gripper to manipulate your sample. And this method is still available at UPenn, but I'm more used to redeposition. All right, let's go to my research part. Because I mentioned that there has been concerns about ion beam damage, so I wanted to investigate whether there is ion beam damage significantly damage battery materials. So this technique, cryogenic lift-out, has been relatively fewer for battery materials. So I'd like to acknowledge some pioneering research using this technique. The first work investigated vitrified SEI by preserving liquid electrolytes. On the other hand, we can also observe interface in even solid electrolytes. Because these research is not compatible with the deposition method, they had to use the lift-out technique to make their samples. And I also wanted to use this technique for my research. And before I really started using this technique, I first investigated ion beam damage to the battery materials. So I used lithium metal battery as a model system, and I studied ion beam damage to them. So as I mentioned, there are two important parts in lithium metal battery. First is lithium, and second part is SCI, surrounds the lithium metal dendrites. And I investigate ion beam damage to each part. So in order to study this, I use commercial lithium metal foils to make TM samples. Because they are not grown in organic electrolyte, they do not form any SCI on there, so that I can purely study ion beam damage to SCI. And I generated cryo-lift-out lithium with two different thickness. One is close to 113 nanometer, and the other one is 250 nanometer. In the meantime, we use xenon ion plasma FIB to study this. I first performed structure analysis using electron diffraction, and what was interesting to me was that they both preserve mainly in the single crystal form of lithium. When you look at the diffraction pattern, regardless of thickness, this means the ion beam does not significantly deform the crystal structures of lithium. However, when you look at the thin sample on the left, you can see clear polycrystalline diffraction rings that can be assigned to double diffraction of lithium oxide. We can also see a six-fold symmetry of lithium oxide patterns, which arise from the texture epitaxy between the lithium and lithium oxide. This texture epitaxy can be seen from the interface between BCC and FCC structures. And surprisingly, and this interface is commonly seen in the interface between the BCC and FCC structures generally. And BCC in this case is lithium, and FCC structure is lithium oxide in our case. And surprisingly, I didn't see such lithium oxide diffraction in the fake sample. Therefore, I hypothesized that there are different ratios between lithium and lithium oxide and two different thickness along beam axis. The following graphical illustration shows my hypothesis from the diffraction observation in the previous slide. I hypothesized that I have two different ratios between lithium and lithium oxide when ion beam sweeps through the reactive lithium. When ion beam sweeps through the lithium, it can create a constant thickness of lithium oxide. And in order to match the diffraction pattern result, I also performed yield analysis, and we found the thin cryo-lift-out has also less lithium oxide along the beam axis when you can see the blue curve, which represents the thin lamella. And when you look at the thicker samples, whose thickness is about 200 nanometer, you can see the lithium KH fine structures is close to the lithium dendrites whose thickness is exactly the same. So this is still consistent with our diffraction observation. So we believe the oxide shell came from the residual oxygen gas in the P-fifth chamber. So to calculate this, we borrow some concept from aluminum diagram where you can predict what temperature the oxidation is thermodynamically favorable or not. We know the partial pressure of the oxygen in the P-fifth chamber, and we know the temperatures, so we can calculate it. According to the calculations, the oxidation is possible if the temperature is below than 2,650 Celsius. Of course, this is under assumption that lithium exists as solid at such temperature, and actually it will melt beyond 200 Celsius. However, what we're interested in the temperatures close to cryogenic temperatures, so our assumption is still pretty accurate. And at the cryogenic temperatures, based on this calculation, the oxidation is always favorable. So that's why we can see some lithium oxide, because lithium oxide reaction. All right, let's go to some interesting part where we can use direct electron detector to detect very subtle and weak diffraction patterns. If you happen to remember the diffraction patterns of the thick lithium lamella, we didn't see lithium oxide diffraction patterns or even see double diffraction, although we did see some lithium oxide in the thin sample. So we have a few hypotheses regarding this observation, but after taking diffraction patterns with metric camera, things got much straightforward to understand. So my question about the diffraction pattern in the previous slide is whether there is lithium oxide in the thick sample or whether it is too weak to detect. So interestingly, when I use a direct electron detector, METRO, we can clearly visualize the lithium oxide diffraction patterns while it was not clear with one view camera. I can also point out the capability of METRO comparing the diffraction pattern between METRO and one view at the same dose rate. And these are the diffraction patterns taken at the same dose rate to emphasize the signal to noise ratio of METRO. I intentionally lowered the dose rate for METRO that is suitable for METRO and switch between METRO and one view to see the diffraction patterns. And you can see how clearly METRO provides diffraction patterns of lithium oxide, and whereas you cannot see such clear signal to noise ratio with one view. Okay, we talk about thermodynamics, and we talk about the lithium oxide diffraction patterns even exist in the thick samples. And we also noticed that the accelerating voltage plays an important role in the oxidation. So as you can see the graph on the left, when we use 5 kV ion beam voltage, the oxidation is extremely suppressed. And I believe the beam-induced local heating can accelerate the kinetics of the oxide formations such that you can see the thicker oxide shell along the beam axis. So the thermodynamics always wants to form lithium oxide in the cryo-fifth chamber, and if you have a certain threshold temperatures, kinetics can be accelerated, and ion beam accelerating voltage plays an important role. And this data also suggests that the oxidations takes place in the P-fifth chamber instead of samples being stored on the liquid nitrogen. Okay, let's go to the ion beam damage to SEI as we talk about ion beam damage to lithium metal. Although there is a lithium oxidation, we can mitigate some issues by reducing ion beam voltage, and we want to also investigate ion beam damage to SEI. To compare the SEI structures, we prepared two different SEIs. One is pristine SEI, where ion beam does not have any influence. We term this as extracted lithium dendrites. And we also prepare another samples by cryogenic lift-out technique. In order to match the same dendrite features, we use the same liquid electrolyte concentration and liquid electrolytes, and we use same capacity and same current density. I first look at the bonding information using cryo-ILs, and the IL spectra between the two turns out to be almost identical, meaning that ion beam does not significantly damage the chemical bonds in the SEI. I zoom in one of the KH, which is carbon KH in this case, and they form both CC bonds and CO bonds from the two different samples, one of which is extracted lithium and the other one is cryogenic lift-out lithium. We can also go for high resolution imaging. The extracted lithium dendrites seem to have layered lithium oxide structures at the edge, while some of the precipitates inside the amorphous matrix, we have the similar structures, lithium layer at the edge and lithium precipitates inside the matrix. The reason why it looks darker is partially because we have redeposition of lithium oxide and copper, which usually occurs in the porous samples, and our dendrite samples are quite porous, so some of splashed redeposition precipitate at the edge of the lithium dendrites, and there was SEI in this region. Instead of a small redeposition issue, now that we're really convinced that ion beam does not damage battery materials, and some of them can be easily what we can mitigate. So we want to apply this technique to real application where high resolution characterization is necessary. In most works, SEI has been always washed by an inner solvent. So we need to wash excess electrolyte, otherwise it is difficult to achieve electron transparency. However, in real batteries, SEIs are always immersed in liquid electrolyte. So there has been question whether SEI is the same before and after washing liquid electrolytes. So what I was wondering is if there is a structure and chemical alteration when you wash in dry electrolyte in the lithium deposits. Therefore, I decided to preserve liquid electrolyte in this case. To observe liquid electrolytes, we use a technique called vitrification. Vitrification is process of the freezing so rapidly that the liquid molecules do not have time to crystallize. So if you put just water molecules in TM column, they will end up evaporating. However, if you cool electrolyte fast, they are vitrified and they do not evaporate in the TM column. When it comes to organic solvents, where solidification points are pretty low, the success of vitrification is pretty high, unlike water molecules. And considering we have high salt concentrations, vitrification points go up. So even if the speed is not too fast, it's not so fast, you can still have high success rates of vitrification. And this way, we can preserve liquid electrolyte in a frozen form inside vacuum, and you can still see features with the presence of the liquid electrolyte in the TM column. So my proposed work is, instead of washing and rinsing excess electrolyte, we can also preserve liquid electrolytes on the copper current collector before you're finally freeze into liquid nitrogen. And once it is vitrified, you can use cryogenic lift-out procedures to thin your samples until you have desired electron transparency. And once you have electron transmission, you can transfer your sample from cryofib stage and cryo-TEM such that you can have high resolution imaging. And in the cryofib microscope, this is how vitrified electrolyte and lithium dendrite look like. You can trench the sample with the preserved liquid electrolyte. You can perform lift out and thinning until you have desired electron transparency. This is one STM image when I prepare cryo-lift-out samples with vitrified electrolytes. You can see the dark contrast from lithium and the bright contrast came from frozen liquid electrolyte. And HDIF imaging shows how we transfer our sample to a cryo-TEM holder, and this shows a HDIF image. Then the HDIF imaging shows that the lithium dendrites and vitrified electrolyte. And because SCI and the vitrified electrolytes are composed of similar constituents, we cannot easily tell where the SCI and vitrified electrolyte is through HDIF imaging. However, when we use ILT, we can clearly see the existence of SCI at the edge of the lithium dendrite. We term the SCI with the vitrified electrolyte as wet SCI here. We found from cryo-ILT that there exists excess lithium salts in the SCI regions when we preserve liquid electrolyte. You can see in the red curve displaying fine structures of lithium KH spectrum of wet SCI. This appears to be a combination of lithium oxide here and lithium TFSI. However, if you prepare another SCI, after rinsing excess electrolyte, the lithium KH spectrum of dry SCI contains only lithium oxide. And this suggests that if we want to perform intake SCI analysis, we may really want to preserve liquid electrolytes. Otherwise, you can readily omit some information there. And in addition to that, by the fact that the salt information can easily disappear, the SCI is quite permeable to liquid electrolyte and solvents. Unlike our previous idea that SCI is complete passivation layer, it turns out the SCI is quite permeable to liquid electrolyte and inner solvents. And this observation is consistent with the recent observation of SCI swelling depending on surrounding environments. We can also perform 4D SEM on the wet SCI to acquire structure information. From 4D SEM, you can get pixelated diffraction patterns in 4 dimensions: real space X, Y, reciprocal space QX and QY. And this certainly provides structure information. And if you have a direct electron detector, we can have 4D data sets with low electron dose. The motivation of using 4D SEM is that the parallel beam illumination has too much electron beam dose, and it can damage vitrified electrolyte very easily. If you have low dose HR, high resolution imaging, you can understand how difficult it is to take one image. So when you change from one magnification to another, sometimes field of view changes. So with low contrast images due to your low beam intensity, it's pretty challenging to locate and navigate your sample. And this is exactly what happens to me. So this the below images shows when I was trying to get high resolution imaging of wet SCI with a direct electron detector metro with those electron dose rates. But while I locate and try to get high resolution image, it turns out it was all damaged. So you can see there is no such spider web structures coming from the vitrified SCI. But after I tried to look at high resolution images there, it happened that I totally damaged the vitrified electrolyte. So in this regard, I believe 4D SEM is really important tool to get some structure informations. This was 2 weeks ago, so there is still plenty of room to increase some resolution in Q space by using nanobeam. So we first perform 4D STEM on wet SEI, and you can see with really low dose rate, you can clearly see the diffraction pattern of lithium dendrites and amorphous structures of the vitrified electrolyte. What our assumption on the wet SEI is there could be some lithium TFSI salt diffraction patterns, but unlike our expectations, they still exist. Majority of SEI were amorphous structures, which means that there are still some amorphous or features in the wet SEI. And because of this, I believe we need to use nanobeam with smaller convergence angle to increase resolution in Q space, such that we can see some bonding information of a low Q space. Otherwise, we have too much amorphous structures, so we cannot resolve such informations for now. What's really interesting, impressive with direct electron detector is we can acquire the inorganic distributions in SEI with low dose rates. So what we can do is locate inorganic compounds distribution in the SEI by virtual dark field imaging from 4D STEM data sets. If you have a direct electron detector, you can acquire structure information with those rates. And unlike the HR high resolution imaging, you don't have to damage your samples while locating and navigating your samples. At low magnification, you can draw a rectangular the rectangular spot where the beam is rested over your region of interest, and you can acquire 4D STEM data with the large field of view. And you can see the lithium oxide distributions. This is the lithium oxide distribution in the dry SEI, and you can see where lithium oxide precipitate in the wet SEI. And you can have more statistical data because it can guarantee large field of view data, as long as your computer has such large data sets storage. All right, this is my end of slide, and I want to thank my advisor, Eric Stach, and co-advisor, Eric Desi. And I would like to thank staff scientists at our university and Think Center, Doug and Jamie. And I couldn't find your image, so I just put a random figure here. And I would like to thank Ben for giving me many helps while I use Metro camera. And thank you for attending my talk.
Yeah, thanks, June. If you have any questions now that you haven't asked yet, feel free to submit those questions now, and we'll start trying to answer some of those questions. So the first question that I have for you, June, is are the SEI layers as beam sensitive as lithium, or which one is more beam sensitive in the microscope?
That's a great question. So it depends how we define SEI. So when it comes to dry SEI, when you fully remove excess liquid electrolyte there, SEI is pretty beam tolerant. But if I were to compare the beam sensitivity between the lithium metal and then SEI, I would say lithium dendrites are more beam tolerant. And recently there has been general consensus about which one is more beam sensitive. And it turns out lithium metal can be observed even at room temperatures because they are metals. They can easily dissipate heat accumulation. So even if electron beam increase the heat there, they can easily mitigate their accumulated heat. However, unlike lithium metal, SEI majority of them have poor heat conductivity, so I believe they can be easily phase transform or beam damage while you go for high resolution imaging of SEI. So that's why we have to use the cryogenic temperatures to go to go for high resolution imaging? So the answer is, I think SEI is more beam sensitive. And if you preserve liquid electrolytes, I observed SEI getting damaged really fast compared to the dry SEI.
Okay, interesting.
Yeah.
Another question that's just come in, how do you transfer the vitrified specimen that's on the cryo holder to the TEM? Do you have some sort of gas, or is it just cold?
Okay, I forgot where it was. So I think that person was asking how I transfer my vitrified sample from
I think once it's in the holder, the TM holder, how do you get it into the microscope?
Oh,
I think
okay. I see. I got it. So let's say we have a TM sample here under liquid nitrogen. It is always inside a cryogenic grid box, which majority of cryobiologists use. So I borrow the concept from the cryobiologist. And the sample is in a cryogrid box, and you can transfer the cryogrid box to the system where we can put the cryo box for the cryo TEM holder. So if you purchase a cryo TEM, cryo transfer TEM holder, there's another working station for the box for the TEM holder where we can handle our half grids, and where we can put our samples to the TEM grid groove. So the answer is we have to use a cryo transfer holder for doing that. And if you wanted to use a dedicated TEM, you can also use that. But I haven't used cryo dedicated one. So for STEM and diffraction, we have to use material science-based TEM, so I have to use cryo TEM holder. So that's how we transfer our sample from the liquid nitrogen to a cryo TEM holder.
Okay.
Yeah.
Great. Then what accelerating voltage did you use for the TEM analysis?
Oh, so I have only used 200 kV. The reason why is I believe radiolysis can be significantly lowered at high accelerating voltage. That's the only issue. And if I change to 80 kV, it's really hassle because I have used the dedicated probe-corrected stem. So if I change to one from another, it will be bother other users. So I have used 200 kV. And another reason why I stick to using the same voltage is the radiolysis. Yeah.
Yeah. Okay.
Yeah, and I use gel on NEOARM for doing that. Yep.
Okay. So someone asks, why do you think the surface oxidation was caused by the sample preparation, I guess in the fib, they mean? They would expect the transfer in the plastic bag before you, I guess that's before you freeze it, to be the oxidizing step.
Okay, I got you. So I didn't pull the data. So in our glow box, where we can store lithium metal foils, there is clear native oxide layer on the lithium metal foil, and if you cut through it, there is a native oxide and passivation layer where further oxidation takes place. There is a pure lithium metal. So if you prepare a cryolift-out lithium lamella, there is a native oxide, and which tends to be 10 nanometer to 50 nanometer. And if you go below the native oxide, there is a lithium oxide. And I would say the native oxide exists here. And if you cut through the samples along the beam axis, you can create an artificial lithium oxide shell because of the ion beam damage. So I can say there is certainly native oxide, but there are majority blocked by the native oxide, so we don't have lithium underneath this native oxide, lithium oxide underneath this lithium oxide.
Okay.
Yep.
What dose did you use for your imaging? What beam dose, electron beam dose?
For imaging, oh yeah, I forgot to put that information.
And I guess
so
there's two pieces to that, I suppose, because I think maybe you showed TEM. Certainly there was diffraction, and then there was STEM imaging.
Yep. So the dose rate for low, how do I say, the dose rate for low dose imaging is around 10 angstrom per 10 electron per angstrom square per second, and the total dose rate is as follows. And for these high resolution TM image, I use quite high dose rate because I use one view. The dose rate is 700 electron per angstrom square per second, and I use pixel time for 0.6 seconds. So it's around, the total dose rate is 300 electron per angstrom square for these images. And in terms of the imaging of wet SEI, I realized that it is really difficult to get high resolution imaging because the electrolyte and wet SEI are very beam sensitive. So even if my dose rate is 10 electron per angstrom square per second, while I navigate or locate my sample will damage very easily. So yep, that's all I.
Yeah. Right. Yeah, because it's of course it's not just the time that you're taking the image that matters. It's all the time where you're trying to find the right place and focus, and yeah, all of that is tricky. I mean, this is where I'm excited about the possibility in the future. You know, can we automate some of these things?
Yeah.
As they do for biology. And you know, single particle cryo-EM, a lot of those steps are really automated so that the only dose that you're giving the sample is the dose rate during the exposure, during the capture.
You're right.
Someday.
Someday, yeah.
Great. So let's see. Okay, I have a question quickly here. If I remember correctly, you told me that you'd never done 40 STEM acquisitions before this project. How easy was it for you to start doing 40 STEM?
How easy? It is hard to answer,
but qualitative question.
Gatan software is pretty easy for me to use. So once you have someone knowledge about how to align beam with 40 STEM, I think you can learn it at once. And the issue is sometimes if I don't put or turn on one view or metro, the dark field image sometimes does not work software wise. So that's one of the issue what I was stuck. So that's why I asked Ben to give me some help. And once he told me, I readily perform 40 STEM. And I'm very new to 40 STEMs. That's why I want to play with nanobeam. And that's another thing I have to do. Yep, that's all I can say.
Yeah, that's okay. Yeah. Yeah, and of course, I suppose the tricky thing about 40 STEM is really the analysis, right?
Yep,
And
yep.
a question just came in just this moment about what sort of processing software
Uh,
you use to analyze the data. I don't know how far you've gotten.
yeah, so if I have a very large data sets with pretty high resolution in Q space, I have to use some software to systematically understand the SEI, the structures in wet
Mhm.
SEI, but I haven't gotten to that level so far. So yeah,
Yeah.
I'm
I
hoping
guess
that
for the dark field image that you showed, I'm
Mhm.
assuming that you did that in digital micrograph.
Yeah,
Is that
yeah.
right?
I use digital micrograph to the virtual dark field image here.
Yeah,
Yeah, I
this,
use the
yeah.
digital micrograph software to put a virtual dark field aperture so that it can
Hmm.
readily show where lithium oxide or other particles distributed in SEI.
Yeah, yeah. Because of course, I mean, this is really basic sort of first analysis that's really easy
Mhm.
to do in our software. But yeah, there's sort of infinite possibilities, I guess, in the 4D data cube. So
Yes.
I look forward to seeing how you can
Yeah,
analyze
sure.
all
Yeah.
that data. Um, let's see, maybe I'll do one or two more questions, and then I think we need to stop here. Um, one question that's just come up. Do you think your cryo workflow is going to be applicable for solid state lithium batteries?
Yeah, sure. I believe um, solid state meta battery will be really idle for performing cryo lift out because sometimes if you want to open coin cells and if you transfer your samples with vitrified electrolyte to um, liquid nitrogen, sometimes they evaporate too fast. So I have to be really quick. But if you have solid electrolyte, I think you can perform, you don't have to worry about this. And another thing is there has been really nice work from UC San Diego group working on characterization of solid electrolytes. Sorry, I don't know where I went. Okay. Working on the solid electrolyte battery using this cryogenic lift-up procedures. And yeah, I believe that solid electrolyte is one of the best way to look at the interface because the interface is pretty defined between the solid electrolyte and lithium metal, unlike organic electrolyte, because organic electrolyte, if your electrolyte are too poor, the interface is there are too many interfaces um, in terms of SEI level. So I would say, yeah, solid electrolyte would be the definitely best way to characterize in this workflow.
Okay. Sorry, one last question as you're on this slide. It's just reminded me of a question from earlier. Um, in this slide, you indicate that ice is condensed on this lift-out sample. Where
Yep.
did the ice crystals come from?
Ice crystals tends to form when you transfer your sample from cryofib and to the liquid nitrogen bath. So once you perform cryo lift out and you have electron transparency, now it's time to transfer your sample to the liquid nitrogen before you finally do cryo TEM analysis. During this step, you have to purge your vacuum tool so that you can take out the sample from the vacuum to the ambient conditions. And during these steps, you have to purge your vacuum transfer tool with dry and liquid nitrogen. At some point, the door for the vacuum transfer will be open to the ambient conditions. At this time, you have a lot of humidity source, So the trace amount of humidity can be magnetized to your samples. So if your shuttle or cryotransfer tool has an anti-contaminator to protect your sample from water condensations, I think that would be ideal. But to my best knowledge, in the Leica microsystems, we don't have anti-contaminator for the material science-based shuttle. So that's where we have a lot of ice concentration. And in the cryobiologist workflow, they have a pre-tilt shuttle. So they have their own anti-contaminator to protect their sample from this. So if you really worry about this ice condensation for your analysis, you can also perform some on-grid TM sample preparation, which is a little bit different from the cryo-lift-up procedure. But yeah, this is where usually ice condensation happens.
Okay.
Yep.
All right. Well, we are out of time. There's still a few more questions we didn't get around to, so we'll try to email you individually with answers to those questions. But thank you all for joining, and thank you again, June, for presenting your work. And yeah, excited about the future results and papers that are coming out of this. So,
Sure. Thank you.
Thank you again.
For having me. Yeah, bye.